try to do this a little quicker. That way you have a few minutes to jump on uh, the STI and U seminar if you are attending today. So again, we'll kind of go over what's due, announcements. We'll kind of discuss the EKG and cardiac cycle, briefly talk over blood vessels, and then talk about RAS, which is one of our hormonal controls for blood pressure. And then I'll mention what the keyword will be for the week. So for general announcements, again, that STI and me or STI and you seminar is this week for extra credit. We have two sessions tonight at 5:30 and on Thursday around 1 p.m. Virtual and then in person tomorrow for extra credit. Dr. Curley is offering 5 extra credit points. So please, if you want some extra credit, you do have to sign up for it. I'll have the QR code on the next slide, and you can go ahead and sign up for that and then just join the session after this if you have some time. For this next couple weeks, biggest events for us are going to be the Unit 2 exam. And then for the hybrid in-person group, you're going to have your first practical next week. We'll talk about the practical stuff later, maybe even next Tuesday. But right now I'll try to focus more on the Unit 2 lecture exam that's coming up. For the STI and U seminar, if you want to take a picture of that QR code that lets you sign up. By signing up, you'll get uh, a link to the video for the virtual session, or also just let us know if you're attending the in-person session. By now, the in-person session might be full. I'm not 100% sure. But the virtual session, if you want to try to attend that, I would take a look at these times and dates and see what works best for you. I'll leave this open for like 5 seconds, but as always, Dr. Curley sent the announcement. You can register there as well. Okay, so very quickly, I'm going to kind of talk a little bit about what we did last week. I won't spend too much in detail. You can look at the Focus Session 3 video for more details on this specific slide. But remember, the cardiac cycle involves 4 different steps. You have atrial systole or atrial contraction. You have atrial diastole or atrial relaxation, ventricular systole or ventricular contraction, and ventricular diastole or ventricular relaxation. What you see in front of you is a brief summary of what each of the steps include. But essentially what happens in the cardiac cycle is that our atrias, both our right and left atrias, those will fill up with blood first, then they'll contract. When they contract, it starts filling blood into the ventricles. Once that contraction ends, the ventricles are theoretically full of blood, and then the ventricles will start contracting. By contracting, it'll start moving the blood up to your lungs or up the aorta throughout the rest of the body. And then once the ventricles are done contracting, your atria or your ventricles are theoretically they're not empty, but they have some leftover blood in them, and then blood will slowly trickle back in to the ventricles through the atria. And then eventually the heart will then start the atrial systole or atrial contraction again. There's a bunch of little key terms that I threw in here. Again, look at Focus Session 3. That kind of went into more detail. I think it's been about maybe 7 minutes talking about this. But I just want to quickly talk about that here. On the right, I did mention I would bring this up this week. So on the right is your signal, your electrical signal. So the heart is controlled by an electrical signal. Primarily, there's 2 nodes. You have the SA node. That is your start to cause the atrial contraction. And then between the SA node and the AV node, there's a bit of a lag, and that allows the atria to fully contract first before the electrical signal gets to the AV node, which then causes the ventricles to start contracting. So the electrical signal for this goes SA node to the AV node, then it goes to your AV bundle, which kind of slows down the signal, and that way um, the ventricular contraction, you kind of see that the signal goes in the center and then goes outward. There's a bit of a lag so that the muscles on the inside of the heart don't contract first before the muscles on the outside of the heart. So by having a lag here, you basically have all the muscle, both the interior and exterior muscle of the heart contracting at the same time. So the signal goes to the AV bundle, then splits into right and left bundle branches, where it speeds up the signal to the Purkinje fibers. And then once the signal gets to the um, end of the Purkinje fibers, then you'll have the actual contraction of the ventricles start. So one of the things I wanted to bring up is kind of how the cardiac cycle connects or matches with the electrocardiogram or EKG, and that's supposed to be ECG, but I misspelled that, so that's my bad. But EKG or EKG or ECG, it's the same. They mean the same, just depending on what country you're in or um, what our academic text has it differently. But essentially, the way that the heart electrical signal will go is you have a quiescence period where you have no electrical signal. It's kind of like a rest period. What I did in this picture here is I added where atrial depolarization and the repolarization and depolarizations and where the systole diastole occurs, along with where your signal kind of triggers. So the P wave is the start of your atrial depolarization or your SA node. So if you remember depolarization and repolarization back from Bio 201, we talked about how muscles need an electrical signal. Depolarization allows all of the ions to cause that electrical signal to cause the muscle to contract. So the SA node will fire off that electrical signal. You have the atrial depolarization, which causes this spike or this little loop that you see here. That is your start of atrial systole or atrial depolarization, and your atria will start contracting. Once that occurs, you have in milliseconds, a very tiny lag before you start going through ventricular depolarization or ventricular systole. And we call this the QRS curve or the QRS complex. One thing to note about this EKG is that you can see atrial systole, ventricular diastole, and ventricular systole in the curve, but you can't see atrial diastole. That could be one of the questions you see on the exam. I know it was on my Bio 2 and 2 exam. And the reason you can't see it is while the atria are relaxing, the electrical signal for the ventricles starts contracting the ventricles. So that electrical signal is much stronger than the atrial diastole signal. So we won't be able to see it on a normal EKG. So what happens here in the QRS complex is that your AV node, which triggers the ventricles to depolarize or to contract, will occur, and that's why you have a large spike in electrical signal. And again, the ventricles they need a much stronger electrical signal to contract because the muscles are bigger and that blood has to go to the rest of the body, whereas the atria are only moving blood from the atria to the ventricles. Once you finish the QRS complex, you kind of have a little bit of like a recovery period for your electrical signal, and then you'll be able to detect the ventricular repolarization. So you have a nice little spike. And then after this spike, or we call it the T wave, you'll have another quiescent period before this loop starts all over again at the P wave via the SA node. Um, if you can describe the EKG by while lapping in the cardiac cycle and the electrical nodes firing, you'll do very well on this test. That's usually one of the things that people struggle with is they can say the cardiac cycle, they can tell us EKG, they can tell us the electrical signal and how that works, but they can't put it together. This is from your notes, which I thought was actually a very good picture, as you can kind of see these items overlap. So you have, atrial diastole, atrial systole, atrial diastole again, and you see ventricular diastole and ventricular systole, and it kind of overlaps with the electrocardiogram signal. Um, the questions I've seen is, hey, where in what part of this curve, so you might be given a picture like this, they might ask you where on this, which point it starts atrial systole. Oh, that's P right here. Or, hey, where does ventricular systole end or ventricular diastole begin? Well, that would be at the end of the T. I think on my exam, I had two questions that were very similar, basically telling me to connect either a picture here, or they'll tell you, hey, for ventricular diastole, what part of the EKG will be shown? What either the P wave, the QRS complex, or the T wave, or something like it's not shown at all, that kind of thing. Again, one more way to visualize this, kind of see I'm focusing on a little bit about this EKG and cardiac cycle. I'd put $5 that you'll get at least a couple questions on this because you can ask a lot of different things on just these two items, tell me parts of the four stages of the cardiac cycle, talk to me about the EKG cycle, talk to me about how they overlap. But there's another way, a good picture from your notes, how to visualize this signal from start to finish. And instead of having the whole signal in one, you can kind of see how the signal starts forming. So I do recommend studying this over. Color coded for contraction or systole, and then relaxation and diastole. Okay, very briefly, I'll go over blood vessels. Blood vessels, for the most part, are fairly simple. Understand that the flow goes from big artery down to smaller artery, then capillaries, and then a medium-sized vein to the big veins. Kind of like highways go to side roads, and then you go into streets and avenues and that kind of thing. So just understand your flow of largest to smallest, and then back to largest from arteries and veins. I do recall a question asking you to put these in order. And so that's a good question to kind of study. One thing we talk about is arterial branching. So why do we not just have one big artery going to all of our tissues? And the reason for that is, it's like having, again, one highway to take us to everywhere we needed to be. It would probably take us forever. The highway would be too confusing. So what atrial branching allows us to do is we go from one big artery to smaller arterioles to then capillaries, and that allows us to get more access to tissues. That allows for more perfusion. That expands your surface area. That also lowers your blood pressure. So the blood pressure at like my fingertips is much lower than the blood pressure coming off my aorta. And by doing that, you wear out your arteries and your arteries less than if you were just have one big artery with a high blood pressure. And again, it kind of allows us to move fluids and solutes. That kind of goes through the more surface area, more perfusion. We can just move stuff more efficiently if we have more roads going to it. If you've probably saw it in the notes, it kind of shows where if you have a cancer cell or a tumor, sometimes your blood vessels will develop and go to the tumor because it doesn't know that it's a tumor, but it knows that the tumor needs more nutrients. So our body will actually make new arteries to feed the tumors, which then make our tumors worse. And that just becomes its own little problem. So arterial branching is very interesting. Whatever arteries you have today may not be the arteries you have later, especially if you work out, if you develop something like a cancer. Your arteries and veins will kind of move around to make sure that they supply the appropriate amount of oxygen and nutrients to your tissues. There are three different types of capillaries. So you have continuous, or what I call those no holes. That is, most of our capillaries are continuous. It supplies most of our tissues. We don't want blood leaking into or leaking out of our arteries and veins. You do have some that are fenestrated, which those have kind of like pores. And so what that allows it to do is in certain places, the blood is able to come out and either get filtered, like in the kidneys, and then it can work its way back into the blood system. And then discontinuous or sinusoids, that basically that capillary end. So it's like the road ending. It's a big open hole. And typically those are locations where blood goes to die or blood become or gets developed. So our bone marrow makes blood cells, so it needs a way to put that blood into our arteries and veins. And the liver is responsible for kind of breaking down some of the cell parts, and so does the spleen. And so the blood basically goes to the liver and the spleen to die and to get broken up into other things that we could use. A couple key terms, we have thoroughfare channels and precapillary sphincters. So what a thoroughfare channel is um, we know that not all blood needs to go to all tissues. So a thoroughfare channel, the basic definition is that it's a road that passes by the other tissues. So I chose to use this image to your right as an example. So the path where the water is on right now is what's called a thoroughfare channel. The water can just go from one point of the farm to the opposite side without splitting off because it doesn't need to go to the grass. Precapillary sphincters, on the other hand, they're kind of like little valves that allow um, some water, some blood to go to that tissue when it needs it. So say you're working out and your muscles are kind of sore and they need blood, the precapillary sphincters might open up to allow more blood to go there. And then once you're recovered and once you're back to homeostasis, the precapillary sphincters could close off because you no longer need blood in that area. Typically, it's those are located in spots where blood flow may be like up and down where you don't need it all the time. Think sitting versus standing, or sitting versus walking. You don't always need blood going to the legs, like as much blood going to the legs when you're sitting as when you're walking. But when you start walking, those precapillary sphincters can open up and provide more blood to your legs, for example. And a lot of our nutrients go through our capillaries via diffusion. So diffusion allows steroid hormones, oxygen, and CO2 to pass through. Um, Other nutrients actually can't go through our arteries or veins without extra help. So things like glucose, electrolytes, those need certain, how do I say, certain gateways to and certain other nutrients to kind of help us absorb glucose or absorb electrolytes. And then things like proteins, they cannot pass through our arteries or veins whatsoever. So you should not have proteins in your blood system. That's usually a bad sign. And you shouldn't be losing anything like that. It's usually a bad sign if you have proteins. That means you have some issues with your arteries or veins. Okay, and hopefully I'm gonna move this out of the way because it's kind of bugging me. Oh, my bad. All right. The last thing I want to talk about is the renin-angiotensin-aldosterone system, or RAS. So there is a unit, I believe the Objective 4, Objective 5 talks about how our blood pressure is controlled. If you realize it's a pretty decently sized chapter, so I would expect this to be on the exam, especially RAS, which is a very intricate blood pressure mechanism. So you have different ways of regulating blood pressure. You have hormonal, you've got neural, you've also got like a local control. But RAS is a hormonal control. And I like to talk about RAS specifically because it kind of ties into something that we learned in unit 1 for the endocrine system. So the scenario that plays out here is that before you even have a low blood pressure, the liver produces something called angiotensinogen, which is a free hormone. It's like an inactive hormone that just flows through your blood system and it's just there for when you need it. It doesn't do anything, doesn't interact with anything, it just stays in your blood system. Say you're walking about and you have a lower blood pressure. When you have that low blood pressure, the kidney will release something called renin. Renin will interact with angiotensinogen and it'll actually create the active hormone angiotensin 1. This hormone doesn't do anything by itself. It actually will flow up to your lungs and it'll interact with something called angiotensin converting enzyme or ACE. By combining with this ACE enzyme, it becomes angiotensin 2. And then angiotensin 2 circulates to the body and it'll help increase your blood pressure using two mechanisms. One, it causes vasoconstriction. So it causes your arteries or your veins to become tighter, which increase your blood pressure. And second, it triggers the adrenal glands to release aldosterone. And if you recall from Unit 1, aldosterone is responsible for sodium retention or salt retention. And so by having aldosterone released in your system, it causes the kidney to increase sodium retention, and water follows sodium in the body. So if the kidney is having us keep more salt in the body or in our arteries and veins, then it's going to cause more water to stay in our arteries and veins, which causes a higher blood pressure. There's other mechanisms that are involved with blood pressure regulation. For for a lot of our body, they take away. We kind of developed in a situation where we don't have like some of the modern stuff that we do now. So for the most part, the body has many different ways to increase our blood pressure. But if you're someone that already has a high blood pressure, there's really only like one mechanism to decrease blood pressure in the body. Because for the most part, the body in a natural living outside without the modern amenities that we have today, we typically never needed to lower our blood pressure. Usually we needed to increase our blood pressure because we're always scavenging for food and running from, whatever it might be. So to kind of tie that back in, and because I'm willing to put $5 that you're going to get at least one question on blood pressure regulation, is to get credit for this one for the hybrid students, just submit two other hormones that help regulate blood pressure, and that could be either to increase blood pressure or decrease blood pressure. Should be straight from your notes, should be fairly simple, but I do want you to review that because again, I'm willing to bet money that you'll have one question, probably two questions on blood pressure regulation via hormones. Besides that, that's really all I have. Does anyone else, or does anyone in the chat have any questions? You can either put it in the chat or you can say it out loud. Um,
I have a question.
yeah, Quinn. So,
Hi, it goes back to depolarization.
okay.
I'm a little confused I understand that calcium um, goes into the cell, right?
Mhm.
Okay, but then it never really talks about it leaving. It just says potassium exits. So when does the calcium like rebalance?
It it may not talk about it, but the calcium does leave. It might be more of, let me see. I don't really have anything on here. Let me stop this recording because